Microgametogenesis. Male gametogenesis is the development of pollen grains, that is a male gametophyte from microspores, technically beginning with the first mitotic division of a single microspore nucleus. The development of pollen grains takes place in anther. Pollen development begins when specialized cells, that is microsporocytes, differentiates in young anther. There are generally hundreds or thousands of microsporocytes per anther, and each passes through meiosis to produce 4 haploid microspores or spore tetrads. As the anther matures and dehydrates, they dissociate from one another and develop into pollen grains. Pollen grains or microspores is the first cell of male gametophyte. It is unicellular, haploid, and its shape varies from oval to polyhedral. Inside each anther, there are several thousands of pollen grains. The pollen grains have the following characteristic feature. Generally, the pollen grains are spherical, about 25 to 50 micrometers in diameter, and mature pollen grain consists of 2 layers, the outer hard layer known as exine and inner thin continuous layer known as intine. Coming to the outer hard exine, this is made up of one of the most resistant organic material known as sporopollenin that enables them to resist high temperatures and action of strong acids and alkali. Further, no enzyme is yet known to degrade sporopollenin, because of which they are well preserved as fossils. The region on exine where sporopollenin is absent are called the germ pores. It helps in formation of pollen tube while the pollen grains germinate on the stigmatic surface. The outer hard exine made up of. Then coming to the next one, inner to the outer hard layer exine is the intine. This is a continuous layer made up of cellulose and pectin. A mature pollen grain consists of 2 cells, a vegetative cell or tube cell. It is larger as compared to the other cells and possesses vacuolated cytoplasm which is rich in reserve food, that is starch, proteins, fats and cell organelles. The nucleus is large and irregular. The second one is the generative cell. It is smaller cell, usually spindle shaped or spherical, with dense cytoplasm and prominent nuclei. The nuclei is large and irregular. Male gametophyte development. It is a process of formation of male gametophyte from a pollen grain or a microspore. The nucleus of pollen grain divides mitotically to form a vegetative nucleus and a generative nucleus. The generative nucleus gets surrounded by cytoplasm to become a generative cell. At this stage, the pollen grain is 2-celled, a larger vegetative cell and a smaller lenticular generative cell. The pollen grains may be discharged from anther at this stage, that is 2-celled stage. However, in some plants, generative cell divides further to give rise to 2 male gametes before pollen grains are shed. These pollen grains are 3-celled at the time of shedding. In most of the plants, 3-celled condition is formed after pollen grain has been shed. The liberated pollen grain germinates on stigma and produce a pollen tube. The pollen tube is covered by intine. It secretes pectinases and other hydrolytic enzymes to create a passage for it in the style. The pollen tube absorbs nourishment from the cells of the style for its growth. The tube nucleus descends to the tip of the pollen tube. The generative cell also passes into it and divides the pollen tube to form two male gametes. Thank you.